Good afternoon, everyone. Myself, Mr. Abhishekh Bhor, PhD Research Scholar, Department of Botany, Professor Ramkrishna More, Arts, Commerce and Science College, Akurdi. Today, I am going to present my research work. Starting with the introduction, as we know, aerobiology is a branch of science that studies the various microorganisms and biologically significant materials present in the air. As we know, the atmosphere of various metropolitan cities is predominating with the biocomponents like pollen grains, fungal spore, and huge amount of dust particles. Nowadays, garbage disposal is a serious problem in various metropolitan cities. Many nearby villages are exposed to the serious health problem due to increased level of aerobiopollutants. Airborne infections are common in such villages. The garbage depot of Moshi is present in the city of Moshi, just 12 km away from the Pimpri-Chinchwad city. Every day, 800 metric tons of waste is dumped at this site by the Pimpri-Chinchwad Municipal Corporation. There is an increasing pressure generated by the resident of this area on the authorities of Pimpri-Chinchwad Municipal Corporation to shift this garbage depot at the other locations. But there is no scientific mechanism available with the municipal corporation as well as with the resident of this area to access the microbial flora of the air and its impact on the human health. The regular air monitoring is necessary to study the microbial flora of the garbage depot. The air monitoring of garbage depot is very rare. Therefore, it was felt that the continuous survey of aerobio components is conducted and it may be helpful to solving or suggesting the solution to the Pimpri-Chinchwad Municipal Corporation and also to the resident of this area. The present study is deals with the assessment of the aero microbiota over the garbage depot of Moshi from January 2019 to December 2019. This is the satellite photograph of garbage depot. It is very near to the Pune-Nashik highway at Moshi. These are some photographs of garbage depot. This is the news article published in the DNA newspaper. Material and methods. The present work is carried out by using Tilak Air Sampler. It is the one of the famous air sampler to study the air. It is volumetric air sampler. It is invented by Mr. Tilak sir. This is the photograph of Tilak Air Sampler. The sample collected by the sampler was analyzed for the identification of spore by these three methods. First is visual identification under microscope. Second one is comparison of the sample with the reference slides. And third is comparison with characteristics of culture colonies. Result and discussion. During this year 2019, from January to December, we collected approximately 30 fungal spores. Out of them 40 belongs sorry 30 fungal spore, 14 pollen grains and four other types of particles. Out of 30 fungal spore, 16 belongs to the Deuteromycotina, 9 belongs to the Ascomycotina and 3 belong to the Zygomycotina. Okay, these are some graphs shows the presence of various fungal spore. The graph shows the percentile concentration of aerobio components on different months of 2019. This is the percentage contribution of each group in the garbage depot. These are some photographs of various fungal spores. So these are some references I have used. I would like to thank the organizer of this conference for providing such opportunity. Thank you.